Okay, so today we have Dr. Vikash Chandra with us. He's a principal investigator at Faculty of Medicine at University of Helsinki, Finland. And his talk today is Candidate Genes to Drug Target for Type 1 Diabetes. A little bit about him. He is a docent in experimental endocrinology and principal investigator at the Faculty of Medicine, University of Helsinki. He completed his PhD from NCCS, Pune, India. Thereafter, he moved to INSERM Paris for postdoctoral training with Raphael Scharfman, where he worked on the first ever established human pancreatic endo-C beta cell line and discovered the novel role of transcription factor RFX6 in mature beta cells. Next, he went to University of Helsinki in Finland for postdoctoral research on stem cell-derived islets and disease modeling with Professor Timo Otansky. And the recent work on the role of T1D candidate gene TYK2 in pancreatic beta cell differentiation and type 1 interferon-mediated signaling has been proposed as a potential interventional therapeutic target for T1D. We have a reference for his recent paper, The Type 1 Diabetes Gene TYK2 Regulates B Cell Development and Its Responses to Interferon Alpha, which came out in 2022 in Nature Communications. I'll drop that in the chat. And thank you so much for joining us, Vikash. We're really looking forward to this talk.
Great. Okay, hello everyone. Good evening from Helsinki. So, as it's really great to. I'm really thankful to have me there in this forum, and it's a great forum. I would say it's a great initiative, and I'm very happy to discuss some recent findings. So my name is Vikash Chandra, and I'm from University of Helsinki, as Monica already said. So today we'll talk about this candidate genes to drug target for type 1 diabetes. So I need not to introduce you about this type 1 diabetes here. So type 1 diabetes is a chronic progressive autoimmune disease and leads to the autoimmune destruction of pancreatic beta cells triggered by poorly defined combination of genetic and environmental factors. So now you can stratify the T1D because it's a chronic progressive disease. So you can stratify the different stages of type 1 diabetes, stage 1 and stage 2, which are non-symptomatic, and then the stage 3 is symptomatic. And the stage 1, you already started the autoimmune process, positive for the 1 year 2 autoantibodies, and then the beta cell mass progressively loss, and you can lose more than 80% of beta cell mass by the stage 3. So what is triggering this type 1 diabetes? Still we don't know. There are big research going on since last decades. What are the reason, which factors are triggering? Definitely genetics, and it's a combination of genetic and environmental factor, because we know that the risks for people to develop T1D in general population is 1:300, but those who have already family member with T1D, the risk is 1 in 20. So you can assume that it's 15 times greater. So definitely there is a genetic relation, genetic factor is there, but it's not all the genetic factors, because you can maybe aware that, and this is a very nice one example in the one of the TrialNet project, that there you find the monozygotic twins who has generally they have concordance rate is around 50%. So genetic link is not entirely responsible for this triggering. So definitely there are other environmental factors that we don't know, but it may be viral infection, maybe socioeconomic situation like your diet and lifestyle and everything, and the important geographical location also. So where you are located. So the combination of these genetics plus environmental factors trigger this disease. If we know some marker, some biomarker, we come up with this for this triggering stage, then definitely it will be very nice for a primary prevention of this type 1 diabetes. But let's more talk about this genetic link. So maybe you are aware about this genome-wide association studies. So this GWAS study had identified several genes associated with the type 1 diabetes. 50% of the genes linked with the one HLA region, but 50% are the different genes associated with the pancreas and PBMCs and pancreas development. So if you can classify it, there are 3 times GWAS when it's a start from 2007 onwards. There is a pre-GWAS and post-GWAS. So pre-GWAS, even since 1974, they already identified 6 genes at least for the just a genetic linkage studies in the genetic inheritance in the family. And then after GWAS, there are a number of candidate genes they identified, like more than 60 candidate genes, non-HLA genes they identified. And then post GWAS also, there are now big analysis like meta-analysis and fine mapping can find there are keep coming the targets. But the problem with this translating these genetic signals to understand the disease mechanism that can help to develop a drug therapy against this disease is really not easy. There are several bottlenecks because majority of these signals are in the non-coding regions. The other thing is that disease-relevant cell type effects, although these are very significant associations, so with the type 1 diabetes. So where we have to look, definitely it has some association in the pancreas and maybe PBMC and the immune cell regulation. So that's also the point when to look and where to look for this genetic signals risk variants. And third and very important is that lack of clinically relevant experiment models. So it's not easy to manipulate the pancreas, pancreatic islets. So for this understanding the disease mechanism, maybe it's in during the development, maybe during the differentiation and other stuff. So these are the bottlenecks, although we have plenty of candidate genes, but to understand, to get the fruit of this GWAS studies, we need a correct model and we have to translate into the mechanism. And moreover, now recent studies also published after GWAS, also now they are trying to combine two powerful approaches like this recent paper in 2021 in the Nature, where they combined two powerful approaches, one GWAS with more than 500,000 individuals and 18,000, I think, T1D patient, and then they combined with the single nucleus ATAC-seq. So they can identify the cell type of specific regulation for the particular risk variant. So this gives maybe more complexity of this candidate genes. So like for example, in this paper, they find that a RS, this genetic variants is associated with the regulation of CFTR genes, which is not previously linked with the type 1 diabetes. And moreover, they found that this regulation is only out of they check on the PBMCs and the pancreatic islet, exocrine, and endocrine. And the interesting thing is that this T1D variant regulates the CFTR in exocrine part only. So not in the other endocrine part, yeah, not in the other cell type. So this is also adding one more complexity that about this genetic signals about this candidate genes that when and where to look. So like this CFTR, so maybe reduced CFTR in exocrine pancreas leads to CF cystic fibrosis related diabetes via intra-islet inflammation and immune infiltration, which can already suggested for the contribute to the T1D, and he also highlight the contribution from the exocrine part for the T1D. So that's I'm telling. So we have lots of information about candidate genes, but translating these genes into the disease mechanism and then developing drug for that. So it's a not easy task. So today I will discuss now to model this one of some of these candidate genes using this stem cell based model. So now there are more uh, uh, well established protocols like Otonkowski lab, our lab has developed already very nice stem cell derived islet differentiation protocols. So I'm going to talk two candidate genes today. One TYK2 tyrosine kinase 2, which is JAK state pathway involved in the JAK state signaling pathway, which is one of the very important cellular pathway in the cells. And I will little bit introduce our this TYK2 paper already we published 1 year before. And the second target I'm going to discuss a little bit is IFIH1. So this IFIH1 is a double standard RNA virus sensor in the cells. It's a part of innate immune system. So this is also very interesting genes. So let's start with the TYK2. So before coming to the actual part, I would like to acknowledge all the people in this study, like for the TYK2 first and Professor Tim Otonkowski, unit head, and then the DECIO from the Brussels for invaluable suggestion during this project. Roberto Malone from Paris for establishing this co-culture assay with the stem cell islets and cytotoxic T cells. And Hajin Ibrahim is the PhD student working mainly on this TIC2 project from our lab. Yoni for the all bioinformatics analysis. We also use lots of human islet co-expression analysis from the Leaf Group lab. Rasmi Prasad and Om Prakash was involved. From Malone lab, Clementine and Frederica also involved for the developing this co-culture assay. And for this IFIH1 project, the PhD student Sophia is mainly working, and we have great collaboration with the Malin in KI for the viral work. So which I will little bit explain to you, and this all the funding agency and stuff.
Vikash, I just wanted to take this moment that offline you
Yeah,
spoke to me earlier, and I asked you directly, how did you
yeah,
get involved into type 1 diabetes research? What prompted that decision? And you shared with me that it was really sort of um, the grant from Anodia, which really prompted you to get more involved with type 1 diabetes. So I just wanted to give a shout out to Anodia and all that they have done. I think it's been excellent, and
excellent. Yeah,
supporting
I
scientists
agree.
like yourself has really, brought more talent into the field.
Yeah, definitely. I really acknowledge Inodia. So when I first come to Timo Lab, so I was on the Inodia grant, and then I joined Inodia. Then my full attention comes to the type 1 diabetes, and there I meet all the T1D expert like Desio, Roberto, and everyone I meet in the Inodia. Means now at least I can say I know most of the people who is working on the T1D. So it's a wonderful opportunity. It's highly appreciated, this consortium,
Great. Just a
and we
little
are
plug to them.
yeah, yeah, it's definitely I have to say. Okay, great. So let's talk about this first gene, STIC2, tyrosine kinase 2. It's a T1D candidate genes. It's a member of JAK family tyrosine kinase. These kinases play a critical role in the intracellular signaling of several cytokines, and particularly this type 1 interferon. And then they phosphorylate the stats, different stat proteins. So just by schematic, if you want to see, so if you come, the interferon comes, so there are different receptors. These are non-receptor tyrosine kinases. So they are in the cytoplasmic domain, and there are four JAK members: JAK1, 2, 3, and JAK4 is actually TIC2. So they are in different combinations for the different cytokine receptor. And like for the example for interferon alpha type 1 interferon, so IFNAR and IFNR2 receptor is there, and that associated with the JAK1 and TIC2. So they are involved for the interferon signaling, type 1 interferon signaling, like for IL-23 is important for the axis for the psoriasis. So JAK2 and TIC2 is there. So there are 7 members of a stat 1, 2, 3, 4, 5A, 5B, and 6. So depending on the phosphorylation and different cytokines, they activate either one or two stats molecules, and then they go to the nucleus and then transcribe the downstream signaling pathway. So the other interesting thing is that about some loss of function, partial, I would say, partial loss of function SNPs in TIC2 associated with the protection against several autoimmune diseases, is already published in 2018 by fine mapping, and earlier also like for the autoimmune disease, including type 1 diabetes and rheumatoid arthritis. So then we also do some homework because we are in Helsinki, and maybe you are aware of that. There is a very great genetic project going on, this FinGen, which is FinGen project is compiled more than 3,000 clinical endpoints obtained from electronic health record data of more than 200,000 Finnish individuals. But right now, in the latest, I think it's more than 500,000 individuals already. Then we take this opportunity and check the SNPs for the in Finnish population, and we find that it's very significantly protecting against several autoimmune diseases like Crohn's disease, rheumatoid arthritis, and psoriasis, including type 1 diabetes. So it was a very good start for us. So we do some more homework to before starting modeling this candidate gene. So one is that it's associated with the protection against type 1 diabetes, but there is no mechanism. We don't understand. We don't know how, and it confer the protection in these individuals who has partial loss of function. So for that, we employed our this beautiful stem cell model, which is just a seven stage model, and we take human pluripotent stem cells to different stage of pancreatic differentiation, like definite endoderm, primitive gut tube. We try to mimic the embryonic development. We go to pancreatic progenitor, endocrine precursors, and then immature and then mature stem cell islets. We already published this protocol, which generate highly efficient and mature functional beta cells in nature biotechnology 2 years before. So also, as I told you before, choosing this candidate genes to model, we do some homework. So we also see that what is the expression level of this TIC2 during the pancreatic lineage differentiation. I would say that during the differentiation, and we are also amazed with the fact that it's continued to express during the course of differentiation. So this is the qPCR for the gene and the protein level. So we thought that it may be possible it also involved in the pancreatic lineage specification, a pancreatic lineage differentiation, because it's continue to upregulate. And since it's an in vitro model, so we also wanted to confirm that. So we are, as I told you, we do several core expression analysis from the fetal pancreas and adult pancreas bulk RNA-seq, and when the first trimester of this days post-conceptions bulk RNA-seq from the 16 fetal pancreas, we also find that the TIC2H continue to upregulate during the earlier stage, which is also the time when the pancreatic lineage differentiation happening. And this is the single cell RNA-seq profile from our differentiation protocol. There are different cluster. You can see this is the alpha cluster, this is different beta cells, this is progenitor and precursor cells. And this is also the adult human islets, alpha and adult human islets beta cells. So it's uniformly expressed everywhere. So after this, we proceed to model these candidate genes because we found it's very interesting. And then we decide to take two questions, like, because it's going to progressively increase during the course of differentiation, does it has a role in the beta cell development? Yeah, differentiation. And the most important thing is that in the mature islet, how it responds to inflammatory signals, particularly type 1 interferon. So for that we generate a knockout model. So what we did, as I saw you earlier, this is a JAK-STAT signaling pathway. So there are maybe more than 50 cytokines and growth hormones which pass through this JAK-STAT signaling pathway. And for example, I here you saw some of the interleukins, some of the cytokines like IL-12, IL-23, type 1 interferon are involved the TIC2 because they have in the cytoplasmic domain TIC2. So when we knock out, these all signalings must stop in the cells. So when you do the type 1 interferon, so you must have lost this activity. To confirm that in iPS cells, we generate the knockout with CRISPR-Cas9, and then we confirm that when we treat these cells, knockout cells with the interferon alpha, in wild type you phosphorylate the STAT1 and the STAT2, but in the knockout, you're not able to phosphorylate the STAT1. And to confirm also that we didn't touch the other receptors, the most important control is that the type 2 interferon gamma, which is composed of on the interferon gamma receptor 1 and 2 in the cytoplasmic domain, you have JAK1 and JAK2, so no TIC2. So you must have survived this, the interferon type 2 signaling, you must survive in these cells. So when we treat with the interferon gamma, you can see that it can phosphorylate the STAT1. So we specifically delete the TIC2 just to confirm this model. And I would also like to highlight that it involves several signaling TIC2. So type 3 is also involved. Type 1 is involved. IL-10, which is anti-inflammatory signaling, that's also involved TIC2. So this we have to keep it in mind that we maybe disturb the several cytokine signaling which pass through the TIC2. So
Yeah,
next
so
we
its activity is sort of contextual, right?
yeah. So next we see that the start differentiating these cells, and then we found that until pancreatic progenitor stage there was no there is no problem until the pancreatic progenitor states both the wild type and knockout cells are differentiating similarly. But at the stage 5 endocrine progenitor stage, we found that there is some impairment in the endocrine precursor formation by this neurogenin, which is one of the marker for the endocrine precursor cells. We found that it's less in the knockout compared to the wild type. And due to this, we found that in the mature final stage, there is a less insulin and NKX 6.1 positive cells. There is also confirmed by the facts. And also the content, insulin content is less in this knockout cell. So there is some impairment in the beta cell differentiation. Definitely it's looking, but they have equal potential. They have equal insulin secreting capacity is not impaired. So we check that. So there is a less insulin in the beta cells, but there is no capacity to secrete insulin is not impaired. And this also we confirm, we always parallelly confirm with the human islet bulk RNA-seq data that we find that neurogenin 3 in the fetal pancreas RNA-seq data that neurogenin 3 is positively regulated with the TIC2. So if you have less TIC2, less neurogenin 3. And in the adult bulk RNA-seq data, we confirm that the insulin and TIC2 has positive correlation. So like less insulin, less TIC2, more insulin, more TIC2 kind. So to understand what could be the mechanism, how it's impaired the insulin beta cell differentiation. So we do the bulk RNA-seq of different stages. And then we found that in stage 5, we particularly found that one target gene. So like here, we confirmed that in the bulk RNA-seq, there is a less neurogenin 3 in the knockout cells and downstream of the neurogenin 3 and KX 2.2. By geo enrichment analysis, we also found that in the knockout, there is a downregulation of beta cell development genes and the regulation of gene expression in beta cells. But when we do the detailed analysis, we found one target KRAS is always upregulated in the knockout. So we confirm by the qPCR and by protein also that KRAS is a target for the TIC2 in this endocrine progenitor and precursor cells. And this we also parallelly again confirm in our bulk RNA-seq from the fetal and adult human pancreas. The KRAS and TIC2 has negative correlation. So if you have less TIC2, high KRAS. If you have high TIC2, less KRAS. And same like for the insulin and KRAS has also negative correlation. So if you have high KRAS, then you have less insulin. So it was very interesting to parallelly confirm in the actual human islet samples. And we also find that the KRAS is important factor in the differentiation of pancreas. If you have high KRAS, you have impairment in the pancreas development in the mice. There are already papers. So then we try to further understand that what KRAS actually doing. So what it's doing in the KRAS, we did single cell RNA-seq, and then the cell type specific pattern of action of KRAS we identify, and then we find that actually KRAS, what it's doing that it's G1 to S phase transition is fast in the process of cell cycle. So if you see in the wild type versus knockout, this is pancreatic progenitor cells, this is cell cycle analysis. By the single cell cycle analysis, you can find the percentage of cells, how much in the G1 phase and S phase. So here we found that in wild type at the same time point, when you have 53% of cells in G1 phase in pancreatic progenitor, in knockout you have 42% of cells in the G1 phase. So it means that KRAS is already shown that it's fast in the G2S transition. And G1 phase is very important for the proper augmentation of neurogenin 3. So this is the mechanism due to this fasten of the cell cycle. Knockout cells doesn't time to proper augmentation of neurogenin 3 that was already shown by Francois Lin in depth cell paper that. So the proper length of G1 phase length is important for the augmentation of neurogenin 3. So we find this mechanism how it's impaired the this beta cell differentiation, but the difference was not very huge that you can see some kind of a absolute a stop of the formation of beta cells, yeah, differentiation in the knockout sample. So this was the one observation. So as I told you, when to look and where to look. So it may have by effect on the different stages of development and differentiation. So, but we are more interested for TIC2 because it's involved the JAK-STAT pathway. We are more interested for the interferon signaling, type 1 interferon signaling. So it's already known that interferon alpha is the key trigger for type 1 diabetes. There are so many papers from the DSU and others shown that interferon alpha crucial in both innate and adaptive immunity and has received increasing attention for its functional contribution to the development of autoimmune response. So we want to understand, and since TIC2 is involved in this type 1 interferon signaling, so we want to study more on this mature islet, stage 7 islets. For that, what we did that we treat those islets wild type and knockout with the interferon alpha for the 24-hour and then go for the single cell RNA-seq so we can understand the cell type specific. So we can understand what happened in the beta cells, what happened in the alpha cells, what happened in the pancreatic precursor cells.
Vikash, before you proceed, we have a question in the audience.
Yeah.
Thank you for your presentation. When you knock down TIC2, does the stem cell derived islet population, in parentheses, alpha to beta cell ratio change?
No, both alpha and beta, I think both, because it's halting the endocrine precursor. So there was no ratio change. I think the both population, the endocrine population is less.
Sorry, they both are affected?
Yeah, both are, because it's disturbing the precursor, endocrine precursor cells. So, yeah.
So, okay, great. Thank you.
Okay. So, yeah, so then we did the interferon treatment for 24 hours, and we are just surprised the fact, and we are just surprised to see the effect that if you see the wild type and wild type plus interferon alpha, you can see that there is a huge upregulation of all interferon signature genes. But in the knockout, it's completely absent. This is the presentation for the top 50 genes, top 40 genes around upregulated, and some 9 or 10 genes downregulated. But it was black and white kind of we are really amazed with the fact that when you remove the TIC2, the interferon signaling is completely inhibited in this. And then we check in the beta cells and alpha cell like separately, and we found that in the left panel, you can see there is a huge upregulation of interferon signature genes, which includes all the MSC class 1, antigen processing and presentation, and other stuff, interferon downstream signaling genes, both in alpha and beta, but in the knockout, it's almost absent. So there is no upregulation of any genes, substantial increase of any interferon signature genes. So it was very surprising that you can completely block this interferon signaling in the when you knock out the TIC2. So it was really interesting. And then you can just to understand that how it's important, this MSC class 1 and TAP, which is the transporter for the antigen presentation. So during the inflammation, this is panel I taken from this journal in diabetes, where you can see in the top panel control human islets and for the expression of MSC class 1 and the transporter associated with the antigen presentation and this insulin. And then this is the T1D human islets, and then the insulin deficient human islets T1D and T1D insulin containing human islets. You can see that how much upregulation of this transporter associated with the antigen presentation and MSC class 1. So then we going to check this all process in this our model. So just in case if I say that, so for the presentation of antigen, you must have develop an immune proteasome. And then after immune proteasome, those peptides, those new novel antigens can come through the transporter. And then it in the ER they loaded on the MSC class 1, and then it represent and then call for the cytotoxic T cells. So like how you start the autoimmunity. So when we check that the constitutive proteasome, the normal proteasome, we didn't find any difference in the interferon alpha minus and plus like PCMB 5, 6, and 7. So there was no difference in the knockout and wild type cells. But when the immunoproteasome, which is responsible during the inflammation, like if you have interferon alpha signaling, so immune proteasome uh, uh, express, and then they start making new novel antigens that leads to present on by MSC class on the cell surface, and autoimmunity happens. So this PCMB 8, 9, 10, you can clearly see that it's upregulated in the wild type, but it's not silent, completely unchanged in the knockout. So immune proteasome. And same for the transporter associated with the antigen processing. So the transporter, so it's upregulate in the, and there are different transporter upregulate in the wild type, but unchanged in the knockout. And then same as I already showed you this MSC class 1. So this A, B, C both are highly upregulated in the wild type, but it's unchanged in the knockout. So it was quite interesting, this finding. And then we found that, so we also want to model this SNP, which I already discussed with you, the SNP which we found in the Finnish population, and it's a very common SNP, which affect their pseudokinase. This is in the pseudokinase domain. And strikingly, this SNP is highly evolutionary conserved and providing protection. And against that, in the Bristol Myers developed an inhibitor for this TYK2 BMS9 at 6165. So this also exactly mimic the same SNP, which is in the highly evolutionary conserved SNP in the population, which affect the pseudokinase domain. So better than modeling knocking this SNP, we just use this inhibitor because it's already published in the Science Translational Medicine paper that this inhibitor is actually mirroring the same evolutionary conserved SNP in the TYK2, which providing protection against several autoimmune disease, while they keep sustain some other signaling pathway to for the avoiding of this immunodeficiency. So this was great for us, and then we started using this inhibitor in our different experiment. So we determined the dose of this inhibitor in our stem cell model, and we use that, and we in the wild type it completely using 1 micromolar TYK2 inhibitor concentration, it can you can stop the phosphostat signaling, the TYK2 activity, and it was highly specific. And in knockout, there is already no signals for the phosphostat 1. So there are two advantages of using this inhibitor. Now we have a chemical inhibitor which can target the TYK2, and moreover, we also mimic the common SNP of this protective SNPs in this model. So we use this SNP, and you can see this is immunocytochemistry for the on the stage 7 islets, stem cell derived islets. You can see the non-treated cells with the insulin and MSC class 1 in the wild type and knockout. And when you treat with interferon alpha, there is a huge MSC upregulation. You can see in the both alpha and beta cells. Here we are staying with just insulin, alpha and beta cells, and knockout, it's completely inhibited. There is no upregulation of MSC class 1. And when we use this inhibitor in wild type, this inhibitor also almost completely inhibited the MSC class 1 expression. In knockout, there is already no upregulation of MSC class 1, is very, very important and good result to see that. And by facts also we confirm these results, and we found that we can using this inhibitor we can inhibit the MSC class 1 presentation. So next we think collaborate with Roberto Malone to see the co-culture as the cytotoxic effect of T cells, whether it's can shown by the cytotoxic T cells. So for that, we establish a new system, co-culture assay with robertomolane. So the hypothesis is that when you have cells and you have interferon alpha in the milieu, so you upregulate the MSC class 1 on the cell surface with the novel antigens, so many different antigens, and then the cytotoxic CD8 T cells come and kill the cells. And when you inhibit this TYK2, you can completely avoid this situation. So for that, we genotype our cells to make a HLA type, matching type co-culture assay. And then after several establishment, we finally able to establish this co-culture assay. And since it has also, TYK2 is also involved in the impairment of beta cell differentiation, yeah, endocrine differentiation. So we take to avoid this confounding effect of the beta cell antigen, yeah, other effect on the expression of antigen in wild type versus knockout. We use this flu reactive CD8 T cell line. So we add the antigen, flu antigen, and then the reactive CD8 T cell, and we use so that we can actually know that and the effect of CD8 T cells on this beta cell islets. And for that, we first treat with the flu peptides, plus and minus, and then we co-culture with the cytotoxic T cell, which are reactive to this flu peptide. So in principle, in hypothesis, if you have a wild type and if you treat with the interferon alpha, this flu peptide go on the surface, and then the cytotoxic T cell will recognize it and kill the cell. And when you have TYK2 inhibitor, it will halt the presentation of this flu peptide on the surface, and CD8 T cells cannot recognize it, and your cells will survive better in this TYK2 inhibitor. So this was our hypothesis. And as I said already, TYK2 inhibitor halt the antigen processing and MSC class 1 presentation. And then the ratio of surviving live versus dead, we can find that the effect of inhibitor on the survival of cells. So this was the setup, and it was interesting that we found that in this experiment at the different effector and target ratio of CD8 T cells, that when you have inhibitor, you have protection against the cytotoxicity from the CD8 T cells. So this was also very encouraging, and that also included in our paper. So although the inhibition of MSC class 1 upregulation by TYK2 inhibitor was complete, but cytotoxicity was only partially inhibited. As I told you, this TYK2 may be involved in several other cytokine signaling, which we don't know. But through this MSC class 1, we can see significant difference between the inhibitor versus non-treated. And then we also found that there is maybe some upregulation of PDL1, yeah, other factors may be involved, but this part of research going on already, what's happened. So yeah, so recently also the recent paper came in Nature Communication that TYK2 signaling promotes the development of autoreactive CD8 T cells in node mouse model. They found that when they use the same inhibitor, which is we use, so they can hold the differentiation of autoreactive CD8 T cells, which is also very encouraging and in line of our research. So that's also support the fact of this managing this inhibitor. So the hypothesis is that where and when to use. So as a secondary prevention, when you know autoantibody positive, this is the window you can use this TYK2 inhibitor to halt the further deterioration of this beta cell mass. And now there are so many JAK inhibitors already available in the market, but with the JAK inhibitor, like this is from the recent review, you can find it. So there are several JAK inhibitors, but we are talking about this TYK2 inhibitor. So you can see that there are several signaling molecules involved through the different JAK components. So TYK2 is involved mainly in the IL-23, 12, and type 1 and type 3 interferon. So when you use this TYK2 inhibitor, you mainly block these two signaling molecule, but you keep open this other signaling pathway so that you cannot compromise lots of your immunodeficiency susceptible to viral other infections. And like for the example, there are baricitinib and others. So and they are blocking the JAK1, JAK2, so you can block most of the signaling pathway related to cytokine and growth factor. Maybe some are the important for your immunodeficiency. So there are different flavors already in the markets. This inhibitor is already in market for already authorized in US and Europe and Australia, I think, and Japan also for the mild to severe this moderate psoriasis, and it's in a tablet form or oral form. It's already working fine. So as I said, the TYK2 inhibition, you need to fine-tune balance between antiviral response versus autoimmunity because you need some immunodeficiency. So that's why we have to find a correct stage, correct dose to get the immunodeficiency versus autoimmunity. Yeah, so in summary, we decipher the unknown role of TYK2 for endocrine commitment in pancreatic differentiation. We provide evidence that perturbation of TYK2 signaling sufficient to protect stem cell islet for interferon alpha response and represents a key interventional therapy for T1D in fully mature beta cells. Although fine dose tuning need to keep the balance between. I think I am already over time, so if you want, I can. Yeah, I can introduce in 2 minutes the new target if you want.
Yes, let's introduce the new target. And then this is a good point from that last slide. If you want to just go back to one quick slide, where you provide evidence that perturbation of TYK2 signaling is sufficient to protect SC islets from the interferon alpha responses. And so I think there was a question earlier in the audience that is, what is the role of TYK2 in alpha versus beta cells if you're talking about the full islet protection?
Yeah, so that's very interesting question. So we found that alpha and beta both has similar antigen processing and representation capacity. So I didn't show you the picture there, but we have same graph, same image from the alpha and beta, which is already in paper. So we hope that it's blocked in both alpha and beta, it works equally.
That's coming out in your new paper.
No, it's already in that paper. So
okay.
Because
okay,
we,
great.
yeah. So it's already
Fine tooth it. Okay, interesting. Yeah, let's hear about the new target. This would be great.
Okay, so I'm sorry I didn't realize that it takes so much time. So now we are working on another very interesting candidate is IFIH1. It's a part of innate immune system, and this is a sensor for double stranded RNA virus in the cells, in beta cells, and it's encode the MDA5 protein, which is intracellular viral sensor. And as I told you, it's part of innate immune system, and also it's a interferon signature genes also. And loss of function mutation have protective phenotype towards type 1D. And if you have gain of function, you predisposed to type 1 diabetes. And partial loss of function shows that you have, like there is a recent paper also that partial loss of function mutation in MDA5 in nod mice protected from a spontaneous and CVV-induced diabetes, where complete loss of function did not. So we need some signaling. So as I told you, this is a sensor in the cells, part of innate immune system. This is a viral double-stranded RNA virus. You know that the most predicted environmental factor is CVV enterovirus, and this is the sensor which first sends the enterovirus in the cells, in the beta cells, and then the downstream signaling start. So this is very important part for the T1D. And one other thing is we found that this is also continue to upregulate in the beta cell differentiation. And another important part is that we found this is highly enriched in the alpha cell compared to beta cells, which is also indicating that alpha cells are more equipped with the better antiviral response, and they are maybe surviving more during the early T1D pathophysiology. So this is one of the genes. So that's why we are trying to model it, this IFIH1. And we have already generated loss of function and gain of function model for this. And loss of function model already showing there is some impairment when we do stimulate with the poly-IC, a viral mimetic infection. So we found a differential inflammatory response in the knockout versus wild type, like for this type 1 interferon beta and CXCL10. And the gain of function was really interesting. when we develop the gain of function, so we are amazed with the fact that when we induce from it's a very rare mutation in the patient, we get it, and this is in the helicase domain. And when we successfully knock in, so we found that even without any stimulation, even the stem cell stage, it's already aggravated the interferon signaling. Like you can see that 60, 40,000 fold CXCL10, even in the stem cells without any induction. And the MDF5, the type 1 interferon, so what's happening here? So even, and we are not able to take further differentiation there. So it means that this sensor is start recognizing, maybe if you have gain of function. So this, I just put a one cartoon from the recent paper where they saw the ADAR versus MDF5 loss of function of ADAR, which mark the self double stranded RNA. So it means that it start recognizing the self RNA, and then it's already putting high interferon signaling. So like there was, if you remember, this was the candidate genes which was identified in the pre-GWAS by just family inheritance studies, and this is one of the first non-synonymous mutation in IFIH1, which was actually the gain of function and which predisposed to the diabetes. So it was really, really interesting, this model. And the important thing is that when we use the TYK2 inhibitor, the interferon signaling is reduced. So, but we are still working on this model, but this is very, very important model to understand how if you have gain of function in the double stranded RNA sensor, how it start recognizing the self RNA and keep boosting the interferon signaling without, as Mary Waldor already published this by the ADAR, which is marking the cell versus non-self RNA and double stranded RNA. So you can assume that how it can regulate even without any induction or any triggering. So if you have gain of function for these kinds of sensor, you can already put very high interferon signaling that can result in the T1D. So we are looking for nice collaboration on this model also. We are also working on, but it would be good if somebody want to work on this model, and we are happy to collaborate and go further on this model. But it's very nice where we have model which can, without any induction, showing this effect. And yes, thank you. This is some old picture in the Automkoski lab. And great, thank you. I'm a little bit over time. Sorry for that.
Great, no worries. So yeah, so this is great. Thank you very much for sharing. And if there's any other questions, if you can drop them into the chat. I had a quick question for you. You know, we kind of touched a little bit on the fact that you know, the there could be some biomarker, you know, possibilities here. And can you just comment on that briefly?
As a biomarker, with TYK2, I don't think that it's work as a biomarker, but it's an important target. But yes, if you find a biomarker for the earlier stages, yeah, triggering stages of diabetes, that would be very nice.
Well, I mean, for the, you know, the SNPs, the TYK2 SNPs, the actual.
Yeah, definitely it can. If you have gain of function, yeah, loss of function, you can have protection. But if you have gain of function, you are predisposed to, you have high interferon signaling, and you're definitely predisposed to diabetes. So if you can consider it as a biomarker in early, but it's already established, we know that. So from GWAS studies.
Yeah, so there's, you know, there's 37 blood biomarkers associated with the tick loss of function. So
Yeah.
how can you go about assembling a signature, is I guess what I'm getting at,
Yeah.
for the certain endotypes, I guess. Is that even a quest to go down, or no?
Yeah, definitely. There are some attempt to this, if I remember, gene score for the T1D. So this could be included, but as I understand that it's not fit with the every cohort and every ethnicity. There maybe needs some improvement, but definitely these are the These are two important marker and important SNPs, which can be considered to include that.
Mhm. Yeah. Well, as you learn more about the delicate balance of the TIC2 and other constituents of this response, then perhaps we'll be able to kind of do some more fine understanding of the SNPs associated with different stages and different pathways to type 1. But
Definitely.
it's a great talk, Vikash. Thank you very much for joining us. And I
Thank you.
hope
Thank
everyone
you.
has a great meeting coming up in Finland in June. And that should be
Yeah.
just a fantastic way to send off Timo to his retirement.
Yeah, we will miss you, but maybe hopefully you can join virtually.
Yes, we
Yeah.
hope to. Thank you so much and have a great rest of your
Great.
day. Bye-bye now.
Yeah, thank you. Bye.